Welcome everyone to Ethics and a Pint through the EugeniJ.McCarthy Center. If you haven't been here yet, I'm Maya from Reagan, the student coordinator that leads politics and a pint in our center. Tonight, as you all know, we'll be discussing human gene editing in the 21st century. Our discussion is led by senior integrated health science major Matthew Burgstahler, and then we have our three panelists. But I'm going to turn over the microphone so that he can introduce them to us. Please enjoy.
Thank you, Maya. Appreciate it. Just by show of hands, who here has heard of clustered, regularly interspaced, short palindromic repeats? Oh, there we go. A couple of hands. How about the less sciencey sounding CRISPR method? How about gene editing? Gene editing? Okay. How about biology? Oh, there we go. Okay, so that's a mutual starting point for everybody. Okay. I'll switch to this one. So a brief confession. When I was agreeing to moderate this event, I said gene editing, fantastic, tons of material, numerous ethical issues, something I'm passionate about. And so I said, okay, I'll do my homework. So I went and I found an MIT video, and the woman said, clustered, repeating, short palindromic, and I said, oh, this is gonna be a long panel. I don't know if I'm ready. The good news is that this panel is not so much about CRISPR as it is about gene editing tonight, and the ethical dilemmas and kind of the topic itself. So if you're unfamiliar with some of these topics, don't worry, we have a panel of people that were gonna help us tonight. And especially help us understand this process of gene editing, how it's talked about in policy, how it might be regulated, and then some of the ethical dimensions of this issue. And so I think really this topic is pretty inspiring and pretty important, because to me it's really where we go in the future as human beings. What is life with and without disease? And also kind of some questions of changing the DNA, the fundamental aspect of of the human race, and then also affecting generations to come, so offspring's offspring, or less sciencey, our children's children. And so some of those ethical dimensions. And so before I get too ahead of myself, I'll introduce myself, a little bit more about the topic and the logistics, and then also our guest this evening. My name is Matthew Burgstahler. I'm a senior integrative health science major. I'm pursuing medicine. I'm a co-director of the Honors Planning and Advisory Council, who's co-sponsoring this event. And this topic, genetics, especially gene editing, I think is extremely important. It's a growing field. It's an exciting topic. Tons of potential, but also it's kind of ripe with these ethical issues, right? So playing God, a supreme human race. What about these marginalized groups in society that make the human race what it is? What would society be like if we were all the same? So some of those issues that I think we'll hopefully address tonight, and hopefully you'll have some questions as well. So overall, the question tonight is gene editing, how far is too far? So I'll take just a moment to introduce our professors who are going to wrestle with these ideas. So first, to my left, Professor Reagan holds a BA in biology from St. Olaf College, a PhD in molecular genetics from Washington University in St. Louis. Professor Reagan's research interests include DNA repair in Baker's yeast, mechanisms and maintenance of genomic stability, and mechanisms of transcriptional control in eukaryotes. He teaches molecular genetics, introductory biology, human biology, and first-year symposium at CSBSJU. So we'll go to him with many of the mechanisms, the sciencey sounding words, anything that he can cover. Also, Professor Friend at the end there holds a BA from University of California, Berkeley, an MS from Missouri State University, a PhD from University of Hawaii. Looking outside, I'm curious. Professor Friend is an assistant professor of politics at CSBSJU. His research interests include international relations, security studies, Asian politics, modern China, international migration and political economy, U.S. foreign and defense policy, health politics and policy, which I think will come to light, and qualitative and quantitative research methods. And then in the middle, we have Professor Herzfeld. She holds a BA also from St. Olaf, degrees in computer science and mathematics from Pennsylvania State University, a PhD in theology from the Graduate Theological Union in Berkeley. She teaches courses in both the Department of Computer Science as well as Department of Theology at CSBSJU, reflecting both of her main interests, being the intersection of religion and technology and religion and conflict. So she writes, studies, and teaches various topics, including computer theory, computer ethics, research in science and dialogue, spirituality and politics in Islam, and research in conflict. So perhaps some of the ethical dilemmas, and also kind of that intersection between the science and the science and the ethics there. So please join me in welcoming all of our panelists this evening. So for tonight, each panelist will have 5 minutes. I have a little smartphone here with a timer. I'll give a 1-minute warning just so everybody has a fair amount of time. And then I will offer a few questions that hopefully bridge together some of these scientific disciplines, these studies. But I also hope that I'll open it up to you for a group discussion. So be thinking of these questions, things you agree with, things you disagree with, questions you'd like to be posed. So I'm excited to learn alongside you this evening, and we'll start first with the biology and Professor Reagan.
All right, thank you. With my PhD in molecular genetics, it's hard for me to be brief on this, but I'll try. So if you think of your genome, your DNA, I always say to people, that's like it's like an instruction manual for how to make a cell. And like an instruction manual, it's written in a language, and the language is composed of letters. So our instruction manuals, if they're written in English, there are 26 letters. You arrange them in different ways. You can make lots of different words, and if you arrange them in the right way, you can make instructions for doing something. DNA works the same way. It's made of letters. There's only 4 of them, called nucleotides, and depending on how you arrange them, you have an instruction manual for making a protein or some other molecule. And in aggregate, in the whole genome, you have the recipe for making a cell. Your genome, the human genome, is about 3 billion nucleotides, 3 billion. Sometimes, if you change 1 letter of that, you can have very serious consequences to the organism. So think of the sentence, the cat ate the rat. The cat ate the rat. Now if I change 1 letter of that, it's the rat ate the rat. Totally different meaning. So there are mutations that people can get that are just that simple. A change of 1 nucleotide, terrible, devastating genetic disease, Tay-Sachs disease, inevitably fatal by age 5. That's a 1 nucleotide change in someone's DNA. A disease like cystic fibrosis is a 3-letter change out of 3 billion. Big consequences to the organism. As well as the changes that make me different from you. Those are just changes in our DNA. More extensive for some than others, but those are just changes in DNA. The reason I look different than you is we have different DNA, in large part. CRISPR is a technique that allows you to change nucleotides. So the cat ate the rat. Suffered a mutation. Now that sentence reads, the rat ate the rat. CRISPR is a technique that allows you to change that 1 R back to the C that it's supposed to be, so that sentence again will read, the cat ate the rat. What's new about CRISPR is so people have been thinking about doing this kind of genome editing. That's called genome editing, by the way, changing that 1 nucleotide from saying rat to saying cat, and thus changing the meaning of that whole sentence. People have been thinking about and working on doing this for a long time. CRISPR is a new technique to do it. It seems quite easy. It seems quite focused. That is, many of these genome editing techniques change lots of different letters in the instruction book of life. CRISPR seems to be able to be targeted very, very efficiently so that you can literally just change that one nucleotide. CRISPR is also cheap and fairly easy, which if you're thinking about doing lots of different things is always a nice positive. And so it's a way to edit the genome, to change nucleotides of the genome on a small scale, like the single letter changing the sentence from the rat ate the rat back to the cat ate the rat. Or maybe changing a few letters, not wholesale changes, not changing me into you.
One more minute.
I got 1 minute, okay. So there are different ways that you can do gene editing. For example, one way that people are talking about is taking white blood cells out of people with cancer, editing the genomes of the white blood cells so that they fight that cancer better, then putting the white blood cells back into people. Quite an uncontroversial application of CRISPR, I think. I don't think really there would be any ethical complications with doing that. On the other hand, people talk about, this is farther off in the future, way farther off in the future, but people talk about being able to change actual genome sequence of cells in the living body. So somehow getting the CRISPR into me and causing it to change my DNA. Again, maybe, well, there are more ethical complications with that. I think the most ethical complications come from the application of taking an in vitro fertilized embryo, changing its DNA in every cell, so that the person that grows up from that embryo, every cell is changed, and they can pass on that change to their children. Changes in the genome that you can pass on to future generations, I think those have the most ethical questions about them. I guess I'll stop there.
That's perfect. Thank you. Thank you very much. We will proceed to Professor Herzfeld with a little bit about her perspective on CRISPR, that balance of kind of the ethics of the debate.
Okay, so do I have to turn this on?
It's on.
It's on?
Yeah.
Okay. So I'm going to pick up where Michael left off and look at some of the ethical issues in a little more detail. When we think about technology, there are two myths that tend to inform our approach. The first is the Promethean myth that in shorthand kind of goes, if we can do it, we should. But there's also the Frankenstein myth, which basically says, don't play God. So the question is CRISPR playing God? And the first thing I would say about this is not really, not in a new way. For one thing, changing the human body is something we've been doing for a long time. The whole field of medicine is about making changes to things that happen in our body. There's also nothing particularly sacred about our genetic code. DNA is not the essence of our human being. And if we look at Christianity, for example, Jesus enjoins his disciples to heal the sick and to help the suffering. And so if this is a technique that gives us new ways of doing that, that's a wonderful thing. Besides this, Genesis says that we're created in the image of God, and, as such, we are also creators. And so there's nothing that goes against religion to say, yes, we should use our faculties to create, to heal, to do all the things we can. But religion also warns us that we tend to overreach. And I think in technology, this is often the case in a sense that we imagine all the good outcomes that are going to come from something. We write all the positive things that are going to happen in our grant proposals, and we tend to not think about negative outcomes the same way. So if we ask the question, what would overreaching be in terms of CRISPR? I think that editing cells, for example, the example Michael gave of taking out immune cells in someone who's suffering from cancer, editing those in vitro, putting them back in a person. This is just a new technique, developing a new medical process. But when it comes to editing the germ cells, is that overreaching? It gets to be more of a question when we say we're going to change the nature of something, whether it's ourselves or whether it's another species, and that change is going to be inherited from generation to generation. Now, why could this be a problem? I think the main problem basically comes down to one of attitude, of purpose. You know, are we using this to cure, or are we really thinking, oh wow, now we can control evolution and we can produce some sort of transhumanist master race? Some of the problems that come up with attitude here, I think the first one is that this tends to make us look at the human being as if we were machines. And we're not machines. We're not just an assemblage of parts and you want to take out the bad part, you know, or put in better parts. There's also a concern that if we end up with a culture that's pressing for perfection, will we not have an increasingly rising bar, always going up and up and up and up? And when do we stop raising that bar? And will we have disdain for people that don't measure up to that bar? Even our own children, will we have expectations of our own children and be upset, saying, hey, wait a minute, I designed you to be a mathematical genius, how come you're not? This could happen. It might also mean that we'll no longer see life as a gift. We'll see it as something of our own making. And I think that becomes a bit problematic. Will we no longer see people who don't quite make it up to the bar of perfection as a gift? Many families who have a Down syndrome child say, I would have never wanted a Down syndrome child, but she's the joy of my life. You know, will we stop seeing that giftedness in life? So I think ultimately we need to proceed with caution. We need to see that genes are not standalone objects, that some bad genes carry with them fitness effects that we might not even know about. We know, for example, that the gene that causes sickle cell anemia also makes people resistant to malaria. Will there not be other things like this that we don't know until we change the gene? So we need to have caution. A couple other reasons to have caution. There might be an ethical outcry against germline therapy, which could endanger therapeutic uses. If people see this as a new eugenics, they might say, we've got to just stop doing CRISPR, at least on human beings, and this might keep us from looking at some of the beneficial therapeutic effects. We also need to ask ourselves, are we smart enough, are we well-meaning enough to guide human evolution? So far, evolution has been guided by nature, by fitness. Will we possibly guide it in a wrong direction? And if we guide the evolution of other beings, you know, like mosquitoes, let's say, that we want to not be able to carry Zika anymore, well, are we speciesist if we tamper with the evolution of other beings for our benefit, but not necessarily for theirs? I think finally, we need to think about the warning that I think was phrased very well by the Russian dissident Alexander Solzhenitsyn when he said, the line between good and evil runs through each human being, and each of our endeavors gets infected by both sides of that line.
Thank you very much. Professor Friend?
Thank you. So there's a lot of really interesting research in science and technology policy regarding biotechnology. Sorry. Yes. I don't want to make out with it. All right, how's this? A little better? Somewhat? Okay. Real close. Okay. This totally threw off my game here. Okay. So the research in particular that I want to talk about today that I find interesting is how policy is being framed, how the issues of biotechnology are being framed in policymaking discussions, and how the average American understands these issues. I think for many in the United States, and I would say around the world, when you talk about biotechnology, they tend to think of a mad scientist using cutting-edge research to create some type of hybrid monster, right? Or designing superhuman babies, something of that nature, right? A lot of what the public understands regarding biotechnology comes from science fiction novels and films, not really journals and books. They think of Gattaca, they think of Blade Runner, they think of Minority Report, all these films that showcase human cloning and replicants and so forth. But within the policy-making discussions, one concept that we often hear is the concept of human dignity. And with CRISPR or other biotech advances, there's this argument that these technologies are an affront to God in the most extreme sense, or at least a threat to the human condition. But the problem is, this concept of human dignity is not often clearly defined. What does it mean? And it's that ambiguity that gives policymakers a lot of freedom to kind of pursue policies that fit their beliefs, religious beliefs, or ethical beliefs. So we often see within these discussions calls to protect the sanctity of human life, arguments that human life should not be commodified or commercialized, that it must be protected, and that the scientist, without any regulation, is a threat to human sanctity in particular. So to give you some example, the Bush administration, right, kind of evoked the image of dignity, human dignity, when arguing that embryonic stem cell research and cloning should be banned, right? That was something that was often referred to, human dignity. But really, the people don't care so much about safety and research ethics, right? They're really tapping into this concept of dignity and the threat to the human condition. So I'm going to read you a quote from Ronald Dworkin, a noted philosopher. And he says, the public isn't terribly worried about safety or research ethics, but instead have some deeper, less articulate ground for that revulsion. Even if they have not, or perhaps cannot fully articulate the ground, they can express it only in heated and logical, inappropriate language, like a bizarre reference to fundamental human rights. Along similar lines, Timothy Caulfield argues that risk and safety are not the issue driving public reaction or public discourse on biotechnology. When asked, the public often lists morality and/or religion as the basis of their objection to many forms of human biotechnology. So kind of policymakers are picking up on this, right? And they're saying that we cannot safely use these technologies without challenging human dignity. On the issue of CRISPR in particular, some policymakers argue that research involving modifying human embryos will open the door to engineering better babies who are stronger, faster, hardier, and more resilient, I quote. So in other words, they're worried about eugenics. They think we're going to create a master race, right? It's the Frankenstein, image to some degree. So we get these kind of two sides of the debate. Some scientists are arguing that this is a good thing and there should be rapid development of this technology, but there are many lay people and the like who are saying that this is a bad thing and it should be banned. And I imagine there are even some scientists on this side as well. But honestly, a complete ban is not realistic. It could prevent research that could be life-saving. It's also easy and cheap and something that cannot be regulated with efficiency. And I'll talk a little bit more about the regulation during the Q&A. So the question becomes, how do we address CRISPR-Cas9 and other biotechnologies in a way that is safe and ethical, and also to kind of raise awareness in the public about what's the purpose of these technologies? Thank you.
Thank you very much. I'm going to actually go right back with a few questions, and then we'll open up for a group discussion afterwards. Hopefully some of these questions touch on something you might be thinking of or feeling at the moment, a question you might have, and then also some questions that hopefully will bridge these different disciplines as well. But I'm going to first go back to Dr. Friend, if I could. Dr. Herzfeld mentioned kind of that it's not playing God, but that we should proceed with caution. And you mentioned kind of in the Q&A, we'll talk about regulatory means. How do we proceed with caution? How is gene regulating going to be done, or how is it done? And then just for the people that aren't maybe of policy background, how is technology normally regulated?
Well, I'll tackle the first one, and if we have time, we can go to the second. So I don't have the answers to this clearly, but there are a lot of arguments out there about how CRISPR, Cas9, or related biotechnologies could or should be regulated. I think many in the scientific community are arguing that this is not a bad thing, that there's a lot of benefit from it. And the first thing we need to do, one of two kind of points would be to foster public trust in the scientific community, right? So that means that scientists should work with bioethics experts to kind of provide information to the community, to kind of dispel a lot of the fears and the kind of science fiction understanding of biotechnology among the average American, let's say. To kind of discuss the ethical, social, and legal implications of human genome modification. So more open discussion, debate, with the goal of raising awareness and, as I mentioned, dispelling kind of misperception. The second thing would be to generate some type of guideline, right, or a set of guidelines. And that would be something that the scientists could work together with policymakers on, and it would be kind of what is ethical and what is not ethical in terms of using CRISPR-Cas9 and similar technologies. So in the case of oversight, this could be done regarding laboratory work, particularly with aims to evaluate the efficacy and of the genome editing technologies. Is this a good thing? Is it being effectively used, or is it being abused in some way? There could be international guidelines established that would, even though they would be difficult to enforce, of course, but they could at least unite lawmakers under some kind of international rubric. And then, of course, making it very clear that no country, and there is work going on in China, in the UK, and in the United States, but no one is trying to create, kind of super babies, right? That's not the intention of the scientists at this point, and it's not really even technologically feasible, at least from what I understand. So those would be kind of the first steps.
Thank you very much. Actually, on that same line, maybe on the question of what is feasible, to Dr. Reagan, when it's talked about in policy, gene editing, perhaps, Dr. Friend mentioned that perhaps there are some misconceptions. If it's politics or politicians, speaking with other politicians, if there's not scientists in the room, there might be a few misconceptions that we're already at the stage of designer babies. Can you give us a little timeline as far as kind of where we are with gene editing, and then also how close are we to making me a little bit taller or better looking? Some of those questions.
Or restoring my hair, that's what I really want. So this, the development of CRISPR has been amazingly fast. Basically, it was discovered about 5 years ago, and has exploded since then as people have seen what a useful technique it can be in the lab. So people are using CRISPR to modify DNA sequences all the time, well, more and more in the lab for various reasons, for experiments that you do on cells that you're keeping in your lab. This is nothing that so far that's been released into the wild or anything like that, but there's been quite a bit of basic work done on how the technique works, trying to increase the efficiency of the technique, trying to eliminate what they call off-target effects. So if you're doing gene editing, you really want to know which nucleotide or nucleotides you're changing, and you don't want to change any others. Those would be off-target effects. So we're trying to eliminate those. But it is used in the lab as a way to edit genes for strictly in lab organisms for lab studies. It has been used on human embryos to change their DNA sequences. These were controlled experiments. The embryos were not implanted into mothers, so no baby was ever born from this, but as a proof of principle, people did take in vitro fertilized embryos, human embryos. They did change the sequence of them. That happened in China, and it probably happened in Oregon. So now that's a very early effort, a very specific kind of proof of principles sort of use of it. But wow, from discovering it to that in 5 years is pretty darn fast. And 5 years from now, I don't know. I can tell you a lot of people are interested in CRISPR. A lot of people are working with it. And scientists are eager to do lots of things with it. There's a keen sense of competition in science. So if we know things are going on in China, and I have a CRISPR lab in the US, I may not want them to beat me, so I may want to get there first, maybe I want to get there right now before they start to regulate me anymore, I don't know. But so that the idea of this, designer babies or making some master race, most of the things that we typically think about, like development of the brain or IQ or things like that, probably many genes go into that. We don't even know how many or how it works. So to talk about screwing around with that at this point, that's probably far off in the future. Something like being able to correct Tay-Sachs, that one single nucleotide, back to what it should be, that's probably closer, I think. But as I say, they're doing this on human embryos right now.
Just a brief follow-up, quick. How close would it be to walking into a doctor's office and having that be part of the experience with a physician in medicine in the future? Would it ever become part of the face of medicine, your genetic code?
Well, I mean, so one of the things that people who think about genomes talk about is the $1,000 genome. When it costs $1,000 to sequence a human genome, then that'll be something that's done routinely, perhaps when you're born, to sequence your genome and know your genome. The cost of sequencing is pretty close to that right now. So knowing your sequence, getting that kind of information about yourself, that's very close. Editing it, that's how long will it be before that's in the doctor's office? Boy, I'm not making any prediction on that.
So I'll turn to Dr. Herzfeld now with this question. Dr. Reagan brought up a few different ways to change the genome, both on an embryo level, so germline versus somatic cells, but then also there's kind of this idea of therapeutic versus disease-ridden versus enhancement, right. And so what where is that line? It seems like sometimes if you were to go a therapeutic route, you might also get the health benefit. If you look a little bit differently, or if you have certain traits, you might feel a little bit better about yourself. You might have a better outcome. Where is this ethical line between disease and the different types of mutations that we could fix?
There isn't one. You know, there's no clear-cut line. A number of ethical scholars draw a line between therapy and enhancement. And they say therapy, that's all good. Enhancement, now you've crossed a line. But as you just pointed out, what actually counts as therapy? You know, we can certainly say, well, Tay-Sachs, Huntington's, you know, all of those, it's quite clearly on the therapy side. But at another point, you know, who knows, Michael, you know, maybe getting your hair back could have a therapeutic effect. So it's going to be as fuzzy a line as it currently is. I mean, currently you have insurers that find it debatable to say, well, where's the line where plastic surgery, for example, is therapeutic, and where is the line where it's simply enhancement? I mean, we already are facing this question in other areas. So there's no clear-cut answer. Another place where people do draw a line is between somatic cell engineering and germline engineering, where they say, well, as long as, you know, if I can somehow, you know, get CRISPR into Michael and he gets his hair back, that's one thing, but his kids are still going to have the gene for male pattern baldness. And they would say, well, if we make a mistake in somatic engineering, we can hurt one person. If we make a mistake in germline engineering, we can hurt whole populations in the future. And I think one of the things that we're missing is that we tend to focus on doing this editing in human beings. But one of the places where this is going to be happening, where there's going to be a big market for using CRISPR to do germline editing, is in like insect populations. And we talk about germ drive, where we would, for example, let's just take mosquitoes, you know, edit them so that they can't carry the Zika virus. And then the whole point is for this to be germline. You want to put them out into the environment and get them to breed with the other mosquitoes so that they put this trait into the entire mosquito population. And, you know, that's a place where we have to ask questions about, are we really smart enough to know what we're doing when we want to put this out into entire insect populations? And we know that those populations, of course, breed very quickly, and so it wouldn't be that many years down the road before the changes could be spreading through the entire population of a given species.
Thank you very much. On that point of marginalized groups, we had a question from the audience. This is kind of directed at anyone who feels so moved. With this technique, would it be possible to eliminate something like Down syndrome? And what would be the ethical dilemmas in your eyes or in society's eyes? Professor Friend?
So that's a great question, and it's actually something that I wanted to follow up with regarding your point of kind of blurry lines, right? So we could think about the list of illnesses that require medical treatment is constantly growing. Certain things we understand to be illnesses today were not always seen as illnesses. And there's a lot of movement in disability advocacy groups that feel under threat. Autism advocacy groups, folks with Down syndrome, small people, the deaf population, they see this as an attempt to...
Could you get just a little bit closer, just for the people in the back? Thank you.
Okay.
Sorry.
So these groups really see their kind of existence, right? It's existential for them. this argument that, oh, we need to remove these illnesses. They even challenge the diagnosis of illness, right? Many in the autism advocacy movement argue that we're not sick. Our brains are wired differently, right? They're kind of embracing neuroscience research. And you now want to remove us from the human population with CRISPR-9 or other related biotechnologies. The same sentiment is held by many groups, right? And this is a challenge to the movement of inclusivity and this notion that we are diverse people and we have different backgrounds and different experiences, and I think it enriches the human condition, and this is a threat to it. If we're going to all of a sudden increase the number of illnesses that could be seen as undesirable and therefore removed from the human condition.
Thank you very much. Well, we had two questions here that kind of covered the same topic, a little bit about privilege. So we mentioned $1,000 maybe to sequence a genome or close to it. That's quite a bit for some people. It's not a lot for others, especially with our interesting insurance market at the moment. So that might be covered, that might not. What do you foresee as any disparities in this price for various CRISPR procedures? And then who would potentially this go to, and who might this disenfranchise?
Yeah, I mean, that there's no doubt that's an issue, as it is with a lot of medical treatments. I have no idea how much it would cost to do CRISPR. But access to this stuff is a big issue, and it's an issue now, though, and you know, we have a system that could be more fair, I'm sure, but we do have a system now, and that system will incorporate these kinds of technologies should they be routinely offered in a medical setting.
Well, there is the concern of classism, right? That certain segments of the population will have access to these technologies and others will not. And that could create, and this may be the kind of more extreme kind of future, but a society with, you know, that has kind of advanced kind of genome and those that have the old-fashioned way kind of thing, right? It's the Gattaca fear, right? And I don't want to play into the science fiction, but I think there is concern about who will have access to these technologies in the future, and I don't think it will be equal access.
I think to some extent, as long as you're talking about editing in vitro fertilization embryos, that will always be far outnumbered by the old-fashioned way of having kids. So until the day comes, I think, when you could, either when society changes so much that IVF becomes the norm, or the day that you can modify in a living human being, I don't think that's a big issue unless one of those things changes.
So I might switch now to a topic, or at least a word that many of you brought up throughout your perspectives, eugenics. It often has that negative connotation, Nazi Germany, other instances throughout history. Where is like the word going almost? Where is eugenics? Is it still a dirty word? Is it still the connotation? Will it be negative forever? Could it ever be a good thing?
I think eugenics is still a dirty word, but not to everybody. You know, in other words, there's a certain segment of the tech-inclined population, I think you see this especially out in Silicon Valley, where they would say, you know, this is not a problem, that actually everything is an engineering problem, and that includes better engineering the human being. But I think that in the general population, there's a great fear of eugenics, and that this partly stems from the past, from not just the Nazi use of this and understanding of this, but even here in America with our experiments on prisoners and things like that back in the early 20th century. It does raise the philosophical question of who gets to decide what would be the master race? Who gets to decide which direction eugenics should go? And that's not a question that has any easy answers.
I would add, I think you're right that indeed it's a dirty word or a stigmatized word in the United States, but not in China, for example, where eugenics research is kind of welcomed and pursued wholeheartedly. It's seen as a good thing, and they don't have the same historical kind of backdrop to work within. So they're making advances as much as they can.
I just would say I'd be in favor of making eugenics a good word again, or making up a new word that doesn't have those negative connotations. There's a program among the Jewish population, particularly up in the Northeast, so there are a number of diseases, genetic diseases that are much more prevalent in the Jewish population. Tay-Sachs is one of those. There is a program by which people can be tested to see if they are carriers for Tay-Sachs. So the only way you can have Tay-Sachs is if both your parents are carriers. They can be tested for Tay-Sachs with when people are they're dating, they're interested maybe in having kids, having a serious relationship, possibly leading to kids, they can be made aware whether they both are carriers or not. And if they are both carriers, they are counseled to take that into serious consideration when they're having children, either the decision to have children or not, or to have children by in vitro fertilization, in which the embryos can be screened for this, and the ones who will develop in Tay-Sachs will not be implanted. The incidence of Tay-Sachs has dropped by 95% in the Jewish population because of that program, which I think is great. And I think that's a eugenics program, I think, and I think it's a good one. It's available to everybody. It's not compulsory. And so those are two key facets of a good eugenics program.
Can I play devil's advocate in a way, right? I agree it's a good thing that Tay-Sachs the numbers are reducing, of course. But there's also this kind of culture now where you need to do what's good for you, what's good for your child, and what's good for society. And therefore, you need to take advantage of these technologies and not give birth to a child that has some disability. There's that kind of, I don't know, ethical obligation almost, moral responsibility that a lot of parents kind of feel this pressure to kind of conform to. So, you know, the line, right?
Yes.
Excellent, thank you. I apologize that we've been keeping the discussion up here. We've taken a few from the audience. By show of hands, does anyone have a question out there that they're curious that hasn't been covered yet? Something on any of these topics? If you feel comfortable, you can raise your hand. If you don't, I can. Thank you. So the question, is it typical for the ethical issues related to a new technology to be the last aspect explored? So does ethics come last in our kind of thoughts about what should we do next in the world? It's either an ethical question or to anybody, I think, really.
Yes.
Only after the fact, right?
Well, you know, to qualify that a little bit, sometimes it's the case that we don't really know what the ethical questions are going to be until the technology gets underway. You know, that issues arise, that there are side effects to a technology that we did not foresee. I mean, hindsight is 20/20, but foresight is not. So we don't always see what the ethical issues will be. However, with some technologies, of course, you can see ethical issues right up front. But I think one of the reasons why the ethical issues come later is that there aren't a lot of research grants out there to study ethical issues. You know, the fact is the money's out there to push the technology, whether it's where the government grants come from, particularly in a place like China, where a lot of the research is government-sponsored, or is in this country too, or whether the research is coming from and done by corporations. The corporations are clearly looking at how is this a money-making proposition, and they're looking at what are the saleable outcomes, what are the good outcomes, and ethics tends then to lag behind because pursuing those questions doesn't make you money.
Any other questions from the audience? Any brave souls out there that are curious about something?
So you said there was 3 billion nucleotides and 4 possibilities for each nucleotide. So by my calculations on my little paper here, that's 81 with 3,861 zeros after it. Possibilities. Do we really want to like go into the possibilities of messing with this, that many possibilities? Because changing one, obviously people are going to decide which nucleotides need to be changed, and there won't be agreement on all of that. But can we possibly understand the repercussions of that many nucleotide changes?
It sounds pretty sciencey.
So the human genome is not a random assemblage of these 4 nucleotides. They're in certain orders. And there's been natural selection. We've evolved, and there's been natural selection to keep some genes basically intact, because when they change, it's usually bad, and the people who have those usually die without leaving offspring, and so those changes are eliminated from the gene pool. So we can be very certain that what causes Tay-Sachs is a 1 nucleotide change, and that if we change it back, the person won't have Tay-Sachs and will probably be just like you or me. I don't think for well-known mutations like that, I don't think there's much danger of unintended effects, because we're changing it back to a version of the gene that most of us already have. The danger comes, the danger, I think more of the danger of unexpected effects comes when you say, this gene seems like it's involved in making, in brain development, and it seems like in mice, if you change it this way, their brains get bigger. So let's do that in humans and see what happens. I think that is an example of one that who the heck knows what would happen if you did that. So correcting mutations back to the versions of the gene that most of us have, I don't think there will be unexpected effects. I wouldn't anticipate that. Doing the kind of thing to change brain development or to give you bigger muscles, those are the things that I think have the greater possibility of having unintended effects. Now, certainly, I think it's certainly true that before anything like that would be attempted, that would be done in model systems, in mice, in apes, who knows, so that we would be as sure as we could be about unintended effects, but of course you never know until you do it in a human.
Thank you, great question. We actually just have about 5 minutes left, a little bit under, for those of you that want to take the 5:15 bus. So we'll try to end on time, but I was gonna go through the panelists again one more time, maybe just a final remark, something to leave us thinking, or any other comment maybe on your thesis this evening. Whoever would like to start.
don't count on scientists to self-regulate on this. If you have an interest in thinking about these issues and guiding policy, I hope you'll get involved, because scientists tend to want to push the envelope. They tend to be competitive, and they will tend to do things unless they get told not to.
I suppose next steps would be thinking about regulation, realizing that these discussions need to continue, and even countries where there's a ban on certain biotechnological practices, India, China, Japan, for example, the ban is not really enforced. So more discussion on kind of regulation, informing the public, more dialogue between various stakeholders, scientists, those concerned with bioethics, the average American, the average citizen, right? Just to have a better understanding of what's going on, and again, to really dispel those fears, to kind of understand the science and the benefit that could be achieved if this was done correctly and pursued in the right manner.
And I would just come back to the quotation that says, if we end up with a society that is pressing for perfection, will we have disdain for the less than perfect? And we already live in a society that pushes towards a perfection that we can never reach. And I think it's important for us to think about that and just think, what are the limits to perfection? And what are the attitudes that we have that make us less than happy with an imperfect world? Because it's always going to be an imperfect world, no matter what technologies we develop.
Thank you very much. So I want to thank you all for attending, and then also please help me thank the panelists this evening. And I'll turn it back over to Maya.
Okay, let's see, is this going to work? Thank you everyone for coming. The next Politics in a Pint will be on November 16th. It will be with Professor Jeffrey Anderson from the Peace Studies Department, and we will be tackling the issue of the oil industry. Hope to see you there.